just want to thank everyone. I can't believe what a pleasure and an honor it is to be here and to be recognized by all of you and to feel the energy in the room last night and today. And you guys are on the front lines. It's a team effort, and we really appreciate everything you guys are doing. I can't tell you how important I think the others. It's a great honor to share the stage also with Dr. Khan and Dr. Shrito, but I think we will all say how important these types of funding is to accelerate the best ideas that we have so that we can get and leverage it for other funding. But this is what drives the innovation, so thank you. And so here's my silly slide. It's great to see the bucket, so I found this on the web. and so I'm going to touch on all three. I think this is a path forward. We need early detection, and I know you guys recognize this. Not everyone does. You guys are really on the forefront. Early detection, we need the proper diagnosis of the disease, and then we need drug therapies. If we can get down that path, then we can get to quality of life throughout life. And that's really what we're after. So today I want to talk to you about some efforts that we're doing on repurposing drugs, existing drugs for Alzheimer's disease, and then tell you what the CART money is going to do to try to advance this further. So we'll start with the bad news. That Alzheimer's, the 145% increase from 2000 to 2017 in the rate of death due to Alzheimer's. Now part of this, as you'll see, is due to the advances in modern medicine. People are living longer, and age is the #1 risk factor for Alzheimer's disease. So if you look at here, this is the rate of two types of cancers over the last combinations of therapies, or are there therapies from these diseases that we might be able to use in Alzheimer's? So we're looking at principles of combination therapies. All of these diseases have succumbed to combination therapies. And are there principles that we can apply to Alzheimer's? And then specifically, are there drugs that we can apply to Alzheimer's to try and help? Now, why has Alzheimer's been so tough? Why is it difficult? Well, part of it is, and this is why I went into neurology, because we all love the brain, but the brain is the most complicated machine that we all know of. I think that's known to any of us. So there's a number of cells in the brain, and they each make about 10,000 connections. So in your brains are about 10 to the 15 connections. And just to give you a perspective, that is the number of hair on the heads of all the people on this planet. That's 10 to the 15. That's how many connections you start off with in your brain. And so it's a Ferrari. You know, it is amazing what it can do, but you know, as it goes on, it kind of breaks down. And so that's what we're seeing, is this is a complicated organ, the most complicated organ that we have, I think, and the most complicated machine. So that's part of the challenge. But also, I think that there's it's a complicated disease, and I'm so glad to hear, like, what you'll see is that there is multiple different flavors of this disease, and I think that's one of the themes. So this is work from Teresa Gomez-Isla. That's my colleague at Mass General in Boston. And she's collected cases where people have come to autopsy, they're cognitively normal, they've come to autopsy for other reasons. And what she shows is that the degree of amyloid plaques. I've told you everyone understands amyloid plaques, so I'm not going to introduce that. But if you have any questions, I should have said this up front. Any questions, feel free to interrupt. Okay, I want this to be a conversation. So amyloid plaques. So here's a patient that's cognitively normal. Here's a patient with Alzheimer's disease. Here are the tangles. Cognitively normal, patient with Alzheimer's disease. So what these cases teach us is that plaques and tangles are not sufficient to evoke cognitive decline. So what is it? So she's been studying these brains. So the difference is neuronal death. That the neurons have to die. So these patients, these are not patients, these are not neurologic patients. These people have plaques and tangles, but they don't have neuronal death. And the other thing they don't have, or that's reduced, is neuroinflammation, is inflammation in the brain. And so this result is, I think, very important. So it says that neuronal death, and particularly the studies show pathologically, the connections, that's why the number of connections correlate with cognition. If you lose connections, you lose cognition. If you lose cells, then you lose the connections. And so that's, I think, one of the things we have to focus on. So this is an example of inflammation in the brain of patients with Alzheimer's. So this is a beautiful collaboration between NIH and pharmaceutical companies, putting their money together, generating tremendous amounts of data, and then sharing it. So that's what we did. We took the data, we downloaded it, and we analyzed it. So what you're looking at here are inflammatory genes, and we looked at different regions of the brain that are affected in Alzheimer's. Red is up, blue is down. So it's pretty clear, red is up. So that there is increased inflammation in the brains of patients with Alzheimer's. Now this was all cases. We took all the cases. And what I'm going to tell you now, and is that we think there are subsets of cases where there's more inflammation. And that not all inflammation is the same, and I think there's going to be multiple paths to Alzheimer's. Just as in today we think about cancer. Like if someone has lung cancer, we don't think of lung cancer as one disease. We think of it as multiple diseases. Someone says you have lung cancer, well, the doctor wants to know what kind of lung cancer, and then the therapy comes. And I think that's what we're going with Alzheimer's, is that there's going to be multiple causes, we need to have the test for it, and then treatments that are tailored towards that. So what we were fortunate enough to do is find, we think, one of the root causes of inflammation in the brain. So there's been lots of inflammation that has been described in Alzheimer's brain, but we think we found one of the root causes of inflammation, and that is this, the name isn't there, it's called double-stranded RNA, but it's a viral, it mimics a virus. It's inside, as I'm going to show you, it's inside brain cells, but we don't think it came from a virus. We think it actually came from, as a consequence of aging, from your own DNA. That each cell accumulates mutations as you age, and then it starts making things that can start to make double-stranded RNA. Now this is a marker, and I'm going to focus on it later, but TDP43 that was discovered by Dr. Trojanowski, one of the reviewers that you mentioned. So you're absolutely right, he's a leader in the field, and we're connecting it up with that, with TDP43. Now, so what's the CART grant going to do? I want to explain to you briefly this technique. So we want to study the inflammation in Alzheimer's brains. We have 1,000 brains in the Mass General Brain Bank, collected by Brad Hyman, and we want to study that. Usually, actually, let me just point this. So what we typically do here is this is one section of tissue, and this is the second section of tissue. So if I want to ask the question, is this the same in the same cell as this? I can't, because they're on different sections. So this technique allows us to go up to, in the brains we're up to 30 markers, in cancer there have been up to 60 markers. So on the same section, so the same cells, we can look at 60 or 30 different markers. So this technique that was developed over at Harvard Medical School by Jerry allows 4 at a time. You take a picture, and then you clear it, 4 more, take a picture, clear it, and then the computer stacks. Where you start with the tumors, and you see all these markers, and then it zeroes in, and it cycles through all the different markers that you can see. So now, just to show you, so here's the inflammatory signature. These are the conventional way we would do. Each one of these are different sections. So here's amyloid plaques, here's tau, here's TDP43, here's the inflammation driver, the root cause. This, because we could do this technique, this is all the same section. So now we can ask, this is a healthy control, so we don't see, but if we look down here, here's the inflammatory marker, we can ask, does it co-localize with tangles? Does it co-localize with A beta? Does it co-localize with TDP43? And what we see is that it's co-localizing with TDP43. It's not in tangle-bearing neurons. So that's important. And now what we want to do with the CART funding is apply markers for the inflammation. So we want, this cell is the root cause, we want to follow that inflammation out. We see it in different regions of the brain. Yes, Norm?
The inflammation, is that swelling? Is that irritation? What is the definition of
So the specific inflammation that we're talking about is an antiviral type of inflammation. It's called interferon. But it's more like it's the type of innate immune system, hardwired immune system that you use to fight off viruses. That's the type.
Okay.
So we can do that now. We want to apply it and study the immune system. Now the other thing we've been able to do is take this root cause of inflammation. So we see it in brains. Is it really driving any disease, or is it just a correlation? So what you see here are human brain cells in culture, and they're labeled green by some genetic trick. And you're seeing the root cause of inflammation, and we put a red dye on it so we can see it. So I hope you can see this, but I'm going to turn on the video. This is over 48 hours, and it's a picture every 3 hours. And what you're going to see is these red dots are going to go into the cells, and then they come down the processes. So neurons are spread out like this. They come down, they surround this part of the cell, and then the cell's going to disappear. You're going to watch the cells die. So if you want, I usually pay attention to the cell here that I pay attention to, and then we'll shift our attention over here. So I'm going to start the movie now. So this is over time. You see that they're coming down and they coalesce around the cell, and now it's gone.
Wow.
Here's another one.
What's happening during that process?
It's down.
What's happening?
So during that process, these cells are brain cells. They mount that whole inflammatory process, and then they die. So that's in 48 hours. So but as a scientist, it's a great, because now we have an assay. We have a way to screen drugs. So here, to Bill's question, here's the cartoon of what's happening. This is the inflammatory pathway, if you will. And in particular, there's a drug that hits this particular part of the pathway. Okay, so then there's actually 3 FDA-approved drugs that hit that part of the pathway. So at low concentrations, what you see here is this is cell death. So normally, if we don't have it there, we're losing about 40% of the neurons. And then as we increase the concentration of the drug, we're rescuing the neurons. And so we see that with these drugs. Now, there's actually hundreds of drugs that target this kinase. And so we've actually tested 10 different drugs that target this kinase, this protein. And 3 work that rescue neuronal death. They're the ones that are FDA approved. There's 7 that don't work. They block this inflammation, but they don't rescue neuronal death. So so now I'll put my doctor hat on and say, okay, we have an opportunity to try to repurpose a drug for Alzheimer's in the patients that have this. We estimate about 50%. As a scientist, you say, great. So we have a drug that works and a drug that doesn't work. This is how we learn something. So the second part of the CART funding is to figure out why are those 2 drugs acting differently. This cartoon I drew up here is too simple because both drugs block this. We know that there's 2,000 genes in this pathway. This cartoon is what, 10, 12? There's 2,000. So we have to figure out we have to look at the others and figure out what's the difference. And the hope is if we do that, then we can define new targets that will be important for neuronal death. The cells in the dishes, just as we talked about, we put the drugs in, and then we use this technique that allows us to do it at 384 wells at a time. And we could still get deep gene expression data. And then we put it through the informatic pipeline. This is what you see. The inflammation signature is here, and this is the signature from the cells that don't have it. And so what we want to see now is with the two different drugs, that we think that they're going to move it more in this direction. We want to know who gets left behind. And so this goes back to detection. So we think that this inflammation, this antiviral type inflammation, is present in about 50% of patients with Alzheimer's. But we have to figure it out in a person, living person, not in, you know, and getting tissue is not an issue, is not a path forward. So fortunately, some of the markers that we've been focused on, we've been able to detect in the spinal fluid of patients. And so we're going to be looking at that. And at Mass General, we've been following patients over time, and we can find that this inflammation is present before people get symptoms. And so we're hoping that that's going to be a path forward. But before that, that's invasive. And because of your interest in early detection, and I confirmed it with Colonel Goldforth that it's okay to discuss this. So I want to tell you just something that's not part of the CART grant, but just that we're also working on. I talked to something about this before, is smell. So smell is also another way to detect Alzheimer's at an early stage. And it's affected early. Now, it's how you process odors that's affected in Alzheimer's. What I teach the residents is, if you know that you have a problem with your sense of smell, as I talked about with some of you, then most likely the problem's in the nose. If the problem's in the brain is when you don't know. You don't know. So most of my patients, if I ask them, they say their sense of smell is normal. But it's affected early, and the olfactory circuit is intimately connected with memory. So the smell part connects right in with memory. And so we've developed a test. It's essentially an odor memory test. It's a memory test, but of odors. And so this is, Sharon, for David, that's the Iowa Hawkeye site. So this is my office, and the Hawkeye fan we were talking last night. So here Sharon is doing it. So this is the device that we use to deliver the odors. We now have it on an app, and we want to be able to do this test in the homes. That's what we're working towards. So the people don't have to come to us, we go to them, and then the information comes to us. So I just want to thank everyone that's been involved in the research. It's a great team at Mass General and at Harvard Medical School that's behind all the work that you've seen and that you're supporting, particularly Steve Rodriguez and Austin Sahan. And then I want to thank our funding sources, and I'm really glad to add that to the slide. So thank you so much for your